Now we're going to look at how the code from DNA will make a protein. And so the genetic code is the basic instructions of how to make a protein. And what we're going to look at is the order of bases in DNA. And every 3 letters or every 3 bases makes up code words. And these code words have a special name, and they're called codons. And every 3 letters will code for a specific amino acid. So amino acids, there's only 20 of them, and they make up the proteins, and the codons code for the amino acids, or it could tell us when to start making a protein, or when to stop making the protein. So, for example, here we have a strand of mRNA from the nucleus. And every 3 letters, you'll notice they've got them blocked off, makes a codon. And so, in order to break this code, you need a decoder. And all of these decoders essentially do the same job, but they may all look a little different. And so, the first step, you start in the middle with the bigger ring, so right here, and you look for your first letter. In our case, we're starting with an A, so we're focusing right here. So that means we're only going to use the bottom left quarter of this circle to figure out our next code. Our second letter in this code is U. So now we're down here under this A still, but we had to go with the U. And our last letter in the code is G. And so we go to this really small piece, and we can see that this codon codes for an amino acid called MET. These are all the abbreviations for it. And so it stands for methionine. But since it's at the beginning of this strand, it also codes for a start codon. So we won't start making a protein until we come across a start codon. So since this is a start codon, we can move on to our next codon, AGA, and decode it. So again, move over to our decoder. And we can see we need to start in this A section again. And so our second letter is G. So now we're up here. And our third letter is A. So that tells you that the amino acid is ARG, or arginine. Our third codon codes for an amino acid that starts with C. So now we're over here. And then our second second base is a U. And then lastly we have a C, which tells us LEU, or leucine. Our fourth codon is UGA. So now we're up top, focusing on this U. And then our second base is a G. And lastly, we have A. So you can see that on this, there are 3 places, 1, 2, 3, where they've got those black dots. Those all represent stop codons. So that means we are done making the protein. We've put all the amino acids that we need to in a line. And so we can stop making the protein. We don't even need to worry about this next codon, because we've reached this stop codon already. So amino acids start with a start codon and end with a stop codon. So this genetic code and this decoder that we've been using, it's the same for all living things. And this shows that all organisms came from a common ancestor a long time ago. Each codon codes for just one amino acid. And this is necessary so that the correct amino acid is always selected. And then most amino acids are encoded by more than one codon. And this reduces the risk of the wrong amino acid being selected if there was a mistake in the code. So it's kind of a fail-safe, like if you made a mistake, it's not necessarily going to destroy the protein and cause problems. So I think we'll have some practice time now with this genetic code.